Explain the prokaryotic cell, and I'm going to start with the outermost organelle into the inside. So I'm going to start with the pili or fimbriae, and these are the little hairs around the capsule. They keep resistance. The capsule, it's the pink and the green smooth part, and what it does is that it keeps from engulfment of eukaryotic cells, and also it tends to cause disease or bacteria. That's why it's considered a virulence factor. Going a level lower, it's a cell wall, and what it does is that it keeps the shape of the cell. Also, it helps to prevent mechanical damage that the cell could receive. What nucleus means, it's nucleus-like, and it's an regular shaped kind of structure. It looks like fibers, and it contains like most of the genetic material, if not all.
I'll be talking to you about the eukaryotic animal cell. The plasma membrane is a membrane that encloses a cell, but similar to the skeleton in our bodies, the cytoskeleton helps to reinforce the cell shape. It functions in cell movement and is made of 3 components: microfilaments, which are flexible but strong, that resist crushing and buckling while supporting the cell. The intermediate filaments provide mechanical support for the plasma membrane when it comes into contact with other cells. These, however, do not participate in cell motility. The microtubules serve as a transport function, as they are the roots upon which organelles move through the cell. These also help during cell division in helping chromosomes to split and move to their new places. The flagellum helps the cell move around. It is composed of a cluster of microtubules within an extension of the plasma membrane. The mitochondria are referred to as the powerhouse of the cell. Their main job is to perform cellular respiration, which means taking nutrients in, breaking them down, and turning them into energy. The centrosome's main function is to help organize microtubules. They consist of 2 centrioles and is important in cell division in that it helps pull chromatids apart. It is important in stabilizing the structure of the cell. The lysosome, also known as suicide sac, it is a digestive organelle where macromolecules are hydrolyzed. They use enzymes to break down proteins, nucleic acids, carbohydrates, and lipids. They also digest the unwanted material throughout the cytosol. They are similar to the functions of a stomach. The cytoplasm is a jelly-like fluid made mostly of water and salts. They contain molecules which are responsible for breaking down waste and also aiding in metabolic activity. Cytosol is a part of the cytoplasm that does not contain organelles, kind of like the blood in our bodies. The Golgi apparatus functions in processing and bundling macromolecules like proteins and lipids as they are synthesized within the cell. This organelle is usually compared to a post office since its main job is to modify, sort, and package proteins to be secreted. It is made of sacs called cisternae. The endoplasmic reticulum has both a rough and smooth ER. The rough ER contains ribosomes, which are the sites of protein synthesis. Smooth ER does not have ribosomes, and it is important in the process of metabolism. Both have to carry various proteins to the Golgi apparatus. It is a transportation system of the cell, just like a factory transporting goods from one place to another. Lastly is the nucleus, which is enclosed by a double membrane that contains perforated pores for material to move in and out of, called the nuclear envelope. The nucleus contains DNA and controls the functions of the entire cell by regulating gene expression, which is why it is called the brain of the cell. The nucleolus within the nucleus rewrites ribosomal RNA and combines it with proteins. The nucleolus is also thought to be the cause of various human diseases. And this concludes the eukaryotic animal cell.
I'm going to explain the plant cell, so I'm going to start with the chloroplast. Chloroplast contains chlorophyll in which photosynthesis takes place, and also the chloroplast transform sun energy into sugars. Last but not least, I'm going to explain the central vacuole, and what it is almost like a little storage bag, and it storages the materials that the plant need and also waste.
The origin of life began with the formation of small organic molecules through abiotic synthesis that eventually turned into protocells that were able to maintain an internal chemistry different from that of their surroundings. As a result, protocells were able to reproduce and metabolize. As environments started to change, protocells then evolved into more complex prokaryotic cells through natural selection. Prokaryotic cells make up the 2 domains of archaea and bacteria. Through endosymbiosis, these prokaryotes were able to come together and form even more complex eukaryotic cells, which make up the domain of eukaryota. Mitochondria is said to have been a separate prokaryotic cell itself, but was engulfed by the eukaryotic cell and eventually became a part of it. These eukaryotes were able to create multicellular organisms like humans, plants, and even dogs. This evolutionary relationship was able to create the world we know today. And this concludes our presentation on active learning module.